Hi everyone, delighted to be here. I was actually a student here. The carpet's much older in 15 years, I can tell you for that. Actually, thank you, Jane, for a great talk there and all the work the office is doing for clinical trials. And what I'm going to talk about today is platform trials or adaptive platform trials. But like everything in life, it's easier to tell a story when you put it into context. And the context and the background for this will be the COVID-19 experience. Platform trials are relatively new. They were used phase 2 trials in cancer to pick the best arm coming forward. There was one trial called I-SPY, a clinical trial on cancer. But actually, REMAP-CAP, which I'll talk about, it's actually the first large platform trial. And platform trials are the new black now. So to see if I get the right one. So some disclosures. So get funded by a load of different people, and did a little bit of consultancy work on pandemic preparedness for Oxford. But the big thing is, the biggest disclosure is, all of the work I'm going to talk about today was done by a huge number of people in a large number of places over a very long period of time. So this is the accumulation of a lot of work by a lot of people. So I'll try and thank some of them as I go along. So, pandemic preparedness, and it was a sort of niche, nerdy thing until quite recently, and unfortunately it rammed itself into public consciousness. And so what I'm going to do is just talk a little bit about how we got here, and some of the things we thought about in the last 7 to 10 years. And the reason I'm doing that is it'll show why a platform or adaptive platform trial was needed, and it'll highlight some of the problems that it aims to solve. Okay, so, and the second thing is I'll talk about REMAP-CAP and explain what all that stands for, and the trial. So, we actually were the crazies. We were the ones saying the end is nigh. And honestly, so I'm an intensive care doctor. If I got in a taxi 5 years ago and said I'm an intensive care doctor, they'd say, did you go to university for that? They'd go, what did you do? Nobody would know what intensive care is, and unfortunately now everybody knows what intensive care is. And likewise, the threat of pandemics, it's actually ever present. And with my friend Steve, he gave a talk in 2019. I think this was in the US Critical Care Society, and was trying to actually highlight that pandemics might be a thing. So the whole thing was, it was the sort of purpose of the intro was to highlight what might cause 1 million human beings to die in a very short period of time. And the top 3 things were, and unfortunately all of these things have got much more likely since 2019, was nuclear war. A nuclear accident wouldn't do it on its own, but a nuclear war would. Collision by an extraterrestrial, like a comet or something. And again, there's been a few near misses recently as well. But the last one was meant to be a pandemic, and a flu pandemic was by far the most common. So if you look back the last 250 years, on average, there's a flu pandemic every 23 years. Okay, so these are common events, and they're getting more common as there's more and more people living with animals, and in their close environment, and air travel. So we got coronavirus, as everybody knows, not the flu, and we all had to change for that. And again, this is sort of what I was getting, is the map on the bottom right is there's a great line from World War Z, the Brad Pitt zombie movie, and it is that Mother Nature is a very effective serial killer. And she is, and she tries all the time. So there are new viruses coming up, generally going from animal hosts to humans, all of the time. Sometimes they're not very effective, it doesn't work. Sometimes they kill people too quickly, so it doesn't spread effectively. But Mother Nature keeps trying. And any one of these individuals can be anywhere in the world within 24 hours by hopping on a plane. So why did we get into this? Why is this intensive care thing, first of all? So it was the H1N1, which was 2009, and I was working in Australia. It started in Mexico and spread around the world and infected probably about a fifth of the world's population. And I was in Australia working with a really good group who do clinical trials. And so everyone knows these now. I mean, these are pandemic curves from the states and territories in Australia. So we collected this information, all the ICUs in Australia and New Zealand, put it together, and it was really useful. But this isn't what we wanted to do. This was plan B. We wanted to do a trial, and a trial of steroids in H1N1. And it was a big question that's still open. Did they work or not? And we were unable to do a trial because by the time these waves had come and gone, we hadn't even got our ethics approval through. And I am not blaming ethics. We had just got our idea together. We had just written a protocol. We were just trying to get some money to fund it. We were trying to get all the approvals and then all the lawyers and the contracts. So we just missed the boat completely. And we, in the whole world, during H1N1 pandemic, under 100 people were randomised into an ICU trial. So we learnt nothing. So the plan was to try and change that. And this is a slide from it's a consortium called Prepare. European Union funded it in 2014. And it ran for 6 years until 2020. And it was to try and change that. So on the left was what we currently do. The first wave, the second wave. Nobody did these curves with 5 waves, which we saw with COVID. And public health response comes out really quick. Preclinical response, the guys and the girls in the labs have the virus and all identified, but the clinical research response was severely lacking. So we wanted to do on the right. We wanted to get the clinical research response out of the blocks quick. What works? What can we treat patients with? So that's why we came up with the idea of an adaptive platform trial. So let me work through some of it. So the RCT is still the gold standard. Okay, so if I have normal community-acquired pneumonia, when I go into hospital, the doctors are going, right, well, what antibiotic should be given? What's the best antibiotic? And there's a number of ones out there. Then to do that trial, you set it all up, you do all the contracts, the ethics, the regulation, you spend years getting the answer, and then you get the answer, and then you go, oh, it would have been really good to know macrolide's another type of antibiotic. It may be synergistically better if you add it or not. You do that trial, take another 7 years. And then again, you take the other trial, and you do it again. And this was sort of the siege mentality, right? And I'm big into clinical trials, and I do loads of RCTs. You sort of go, oh, I can probably only do about 5 of these in my professional career because it takes so long and it's so much work. And you get one answer, and then you have to move on to the next one. So it's not very efficient. So this is the brief overview, but I'll go through it in more detail. So what if you could do things a bit different? Again, maybe another thing to point out about the traditional RCT, at the start, you have to sit down, you get the inclusion criteria, you get the exclusion criteria, you get your endpoint, you say, okay, do the stats, it's going to take 1,000 patients to answer this question, and we're going to stop halfway, go to our DSMB, and they might give us a steer or tell us if we're causing harm or causing real good. But you're locked in, you've bought your timeshare apartment, you've signed the contract, you're in. The REMAP aims to address a few of those things. So REMAP stands for Randomised, Embedded, Multifactorial, Adaptive Platform. And I'll go through all those things in detail. But essentially, you have an eligibility criteria, so you find your patients. You can actually randomise to multiple interventions at once. So one patient can get an antibiotic, a long duration of say, macrolide, and no steroid, let's say. And then you measure the outcomes, but you feed this information back in as you go. Okay? So you're learning as you go, and also you have the ability to change some of these things as you go. And we thought these would be particularly useful in the context of a new pandemic. So let me go through a few of these elements. Now, this trial worked out well, so I'll show you some examples, and we're all very happy and it was all great. But we were the problem child for a long time. This was a 7-year program, 7 years of funding, and it took us 5 years to figure out all the logistics and the planning. So there is a lot of preparation that goes into setting up an adaptive platform trial. Now, this was the first large clinical one done, so it won't be as hard in the future, and we're sharing all the stuff we've set up, but there was an awful lot of planning on we'll be able to know well all the documents. There's a core protocol, there's appendices, there's statistical appendices. We had to work out what disease we wanted. We had to do loads of simulations to see how the trial would perform. We had to plan all of this in advance, and then we built in a sleeping pandemic bit as well, which I'll explain this in a second. But just to say, there's an awful lot of pre-planning. I think the world is learning, and that's becoming quicker. But I wouldn't want to give you the impression these are quick, easy things to do. And the guy Herman Goossens from Belgium, who led the whole consortium, there were 12 work packages, and our one was the slow one that was eking along. It just worked in the end, but there's a lot of work involved setting it up. So, what did we pick? Well, we picked pneumonia. So pneumonia in the ICU is it's a big thing in of itself. Unfortunately, every winter, loads of patients are admitted with pneumonia that require either ventilation or drugs to keep their blood pressure up while we treat them. There are loads of things we don't know, and it would account for about a quarter of ICU patients. And it is, I think, the 4th biggest killer in the world. So in of itself, that is important. That is a disease we need to know a lot about. But the reason we picked that was when a new viral pneumonia, we thought influenza, comes along, the canary in the coal mine is the ICUs. People start popping up with a viral pneumonia of unknown etiology, and that's what happened with H1N1, and that's what happened, I think, when we saw when Italy started just to get hammered as well. That the design of this trial was we could pivot and actually start randomizing patients with this new unknown viral pneumonia. So, and one of the interesting things about these platform trials is it's sort of set up as a perpetual trial. It will continue to answer questions in community-acquired pneumonia, and it will continue to be ready for the next novel pathogen. So we set our inclusion-exclusion criteria for community-acquired pneumonia, but then we set additional inclusion criteria for a new pandemic. But that was all approved in the background, but sleeping. Then, so observational studies are super, and we do some of them as well, so I'm not putting them down, but they show things that might work, but they're hypothesis generating. They don't actually generate the evidence we need to guide our management. You need to randomize patients, so the R in REMAP-CAP is for randomization. But it does this thing called response adaptive randomization, which was pretty new with some amazing statisticians in the US, Berry Consultants. And essentially, you know that most trials randomize a patient 1-to-1. So you get no steroids or you get steroids. Well, each time the model churns, it learns a bit, and it allows it to go, I think steroids, let's just say, are better. So I'm going to randomize more patients to steroids. And then it learns again and goes, okay, steroids are looking pretty good. It randomizes more people to steroids again. The advantage of that is it's probably more ethical, we thought, because a patient doesn't have to wait the 7 years of the trial to benefit from the results. Patients can benefit earlier on in the trial. Now, there's things we're learning that if there's big swings, it can affect the power of the study. And there's things we have learned in this that we will put guardrails on response adaptive randomization, so we don't allow large swings. But again, really novel, really interesting. Embedded, for these things to work, it has to be just part of the day-to-day ICU. And we did a lot. These are open label studies, so no placebo. And it was designed that way because in the context of a pandemic, it's going to be very hard to do a placebo-controlled trial. And we try and embed it as much as possible into the ICU so that patients are identified on the routine screening when they're admitted with a query pneumonia, and that the treatments are written up on the normal drug chart where the treatments are written up. So as much of the trial processes are part of the routine care. And we went through a lot of this with the regulators and the ethics committees as well. So the multifactorial bit, so in fairness, it's pretty neat. So like there's 3 examples there of, I said steroids, antibiotics, and that macrolide thing. So one patient can get actually randomized to a regimen. So, like this. So domain A is the antibiotics. So one of the antibiotics, domain B is the macrolide, long duration of macrolide, and domain C is the steroid. But actually it goes way out past C. Patients can be randomized to multiple interventions at once. And this has allowed us to do 2 things. As we get an answer, we incorporate that into the standard of care. It drops off, but we can add in more and more things. So actually, over the course of the pandemic, there's been 63 interventions added into the trial. It also allows us to do one other really neat thing, is to work out actually the antibiotic and steroids together are much better than either on its own. So actually, the domains can interact. So the beast churns along, collects data, they go to regional data centres, they get all merged centrally, and we don't, at the bottom right hand corner, we don't have a predefined sample size at the start. We have statistical triggers. So if something is going to be superior, so it's over a 99% chance it's the best. If something is going to be inferior, under a 1% chance it's going to be best, or is something equivalent, and we just said that it's a 90% of less than an odds ratio of 1.2. So we had those statistical triggers, and the trial recruits and churns on. And if it hits one of these triggers, it pops up, and then we stop that intervention. We make a decision, we stop the intervention, and then unblind the results and analyse it. To do all this, there's an independent statistical analysis committee that do all these analyses. And again, as I'll show you when we go through it, the steering committee can interfere with this in the sense that if external evidence comes in, we can just stop interventions and publish the results. So that's sort of what it is, but let me just tell you how it worked. Right. So this is from yesterday. So this places all over the world. The top left, so actually one below it. So there's been 12,285 patients randomized into this trial. Of those 12,000, they've received 21,000 interventions. So like nearly most people got 2 interventions. Okay, and then there's 61 interventions have been added in various regions, because it doesn't have to be across the whole platform. It can just be in some places, and 325 sites involved. So the peacetime model is what we set up. So as everyone here knows, it's hard to set up a trial. You have to do all the contracts and all the bits. So we set up antibiotics. It doesn't really matter what they were, but just 4 different antibiotics to test, and that's still ongoing. There's over 1,000 patients in that. The macrolide, big question, is 3 to 5 days better than 14 days? We still don't know. Ongoing. The steroid question, I'll pause here for a second, because steroids is a big question in pneumonia, and we're actually dealing with this now. There's a New England Journal paper out last week saying the steroids might be good in community-acquired pneumonia, but this is the first time external evidence has come out. So we set it up so you either get no steroids, you get a fixed duration, or you get steroids until your shock resolves. And that was running during peacetime, and the whole idea was that we can actually just roll that out when the pandemic came. So, the next day we could just start it. And ventilation, a mechanical ventilation arm as well. So, the war arrived. The recovery trial, which was a UK trial, showed in 4 months, pretty amazing, but at mainly ward level, that steroids were better. So we then, we had, the day the WHO declared COVID a public health emergency of international concern, we started randomizing people into the sleeping steroid arm. We were able just to switch over. And we picked a pandemic 21-day outcome of 21-day mortality or organ failure, because we needed to get answers quickly. We couldn't wait for our normal 90-day survival. Red, dark red means dead, unfortunately. Letter red means organ failures, and then the blue means less organ failures. So more red is bad. So more red with no steroids. So we found the same result as recovery. We found it in a different class of drugs. We used hydrocortisone, they used dexamethasone. That was important during the pandemic because we were running out of drugs. And we showed it in ICU patients. So this then became incorporated the day it was published into the WHO, and then a few months later into the HSE guidelines. Everyone was getting hydroxychloroquine. It was the Wild West at the start of the pandemic. Everyone was trying everything. And again, the recovery trials showed that hydroxychloroquine wasn't beneficial in ward patients. We actually showed so we stopped the trial, analysed the results. We didn't hit a trigger because we stopped early, but we analysed the results because we always err on safety. What we found was hydroxychloroquine caused harm, and combinations with a HIV drug, which people were using, caused harm too. So this ended the practice of giving hydroxychloroquine in ICU patients. So, and then immunomodulation. So this was the first one that REMAP-CAP showed itself. So it actually hit a trigger. So these were drugs for rheumatoid arthritis that dampen down the immune system. And the problem is, in COVID, it's your body. We were not designed to survive ICU. Evolution has got us to, was more interested in the caveman. And these anti-inflammatory drugs were actually beneficial. So they reduced mortality by about 10%, and they reduced the length of stay by about 10 days, which is really important in a pandemic. So this was the first trial to show this, and then again, it became part of the guidelines, and subsequent trials showed the same results. Anticoagulation. So this is interesting for a different story. So COVID causes clots. Hey, if I give people blood thinners, it'll stop clots. Maybe that's good. So we did that in patients with ICU, and therapeutic dose anticoagulation, more red. So actually it was causing harm. So that stopped that. But interestingly, we did it in ward patients too. So the REMAP-CAP was trying to expand beyond the walls of the ICU into the wards. So there's much less red because ward patients are much less likely to die. Your mortality is much higher in ICU. But we find the opposite. Therapeutic anticoagulation was beneficial in ward patients. So really interesting, we find there's a differential effect. The same treatment can be harmful in ICU but helpful in the wards. So we can't lump all the patients into one group. There's actually different effects. And if we had maybe done all of these patients as one, we might have found no result rather than actually two different signals. Convalescent plasma, we didn't do that in Ireland, but we did it in the UK. It was a Herculean effort. They got blood from patients who had COVID, they spun it down, they got the antibodies out, and they gave it to people who had COVID. It was always thought that this would be the absolute best treatment ever for a new pandemic. It didn't work. And it cost a fortune. Right? And I'll skip the antiplatelets. This slide is just to say one thing. We did a 21-day outcome. We based our results off it, we published it, we based the guidelines off it, but then we followed all the patients up to 90 days, so long term, and the results were the same. So actually our 21-day outcome was the same as the 90-day outcome. So we can have pretty good faith in the advice we were given was the right advice. There was a lot of nervousness when we did this. So just to say, platform trials are really good. They're a pre-existing framework. It takes a lot of effort. You can do all the ethical and pre-approvals, well, a lot of them in advance. The subsequent approvals are quicker because we've already agreed 90% of the things Jane talked about in her slide. It's just the intervention is the new bit. And we have the flexibility to add new things in as we learn, which is important. So the trial did exactly what it said in the tin. And the big thing is we have to get ready for the next pandemic because there'll be another one. And it's working together with the community, including the research and ethics committees, the ICU staff and patients' families to learn how we do these trials and how people can have faith in the results. And I think that's it. Thank you.